Microbes in human welfare. Beside macroscopic plant and animal, microbes are the major component of biological system on Earth. You have studied about the diversity of living organisms in class 11. Do you remember which kingdom among the living organisms contain microorganisms, which are the ones that are only microscopic? Microbes are present everywhere in soil, water, air, inside our bodies and that of other animals and plants. They are present even at sites where no other life form could possibly exist. Sites such as deep inside the geysers thermal vent, where the temperature may be as high as 100 degrees centigrade, deep in the soil under the layer of snow several meters thick, and in highly acidic environment. Microbes are diverse protozoa, bacteria, fungi, and microscopic animal and plant virus, virions, and also prions that are proteinaceous infectious agents. Some of the microbes are shown in figure. Microbes like bacteria and many fungi can grow on nutritive media to form colonies that can be seen with the naked eye. Such cultures are useful in studies on microorganisms. In chapter 8, you have read that microbes cause a large number of diseases in human beings. They also cause disease in animal and plant. But this should not make you think that all microbes are harmful. Several microbes are useful to humans in diverse ways. Some of the most important contributions of microbes to human welfare are discussed in this chapter. Microbes in household products. You would be surprised to know that we use microbes or products derived from them every day. A common example is the production of curd from milk. Microorganisms such as lactobacillus and other commonly called lactic acid bacteria grow in milk and convert it to curd. During growth, the larvae produce acid that coagulate and partially digest the milk protein. A small amount of curd added to the fresh milk in inoculum or starter contain millions of larvae which at suitable temperature multiply, thus converting milk to curd, which also improve its nutritional quality by increasing vitamin B12. In our stomach too, the larvae play a very beneficial role in checking disease-causing microbes. The duct, which is used for making food such as dosa and idli, is also fermented by bacteria. The puffed up appearance of the duct is due to the production of carbon dioxide gas. Can you tell which metabolic pathway is taking place resulting in the formation of carbon dioxide? Where do you think the bacteria for the fermentation come from? Similarly, the dough which is used for making bread is fermented using baker's yeast, Saccharomyces cerevisiae. A number of traditional drinks and food are also made by fermentation by the microbe. Toddy, a traditional drink of some part of the southern India, is made by fermenting sap from palm. Microbes are also used to ferment fish, soybean and bamboo shoot to make food. Cheese is one of the oldest food items in which microbes were used. Different variety of cheese are known by their characteristics, texture, flavor and taste, and the specificity coming from the microbes used. For example, the large hole in Swiss cheese are due to production of a large amount of carbon dioxide by bacterium named Propionibacterium shermani. The Rocky Halt cheese are reactant by growing a specific fungi on them which give them a particular flavor. Microbes in industrial product. Even in industry, microbes are used to synthesize a number of products valuable to human beings. Beverage and antibiotics are some examples. Production and industrial scale require growing microbes in very large vessels called fermenter. Fermented beverage. Microbes, especially yeast, have been used from time immemorial for the production of beverages like wine, beer, whiskey, brandy or rum. For this purpose, the same yeast Saccharomyces cerevisiae used for bread making and commonly called beverage yeast is used for fermenting malted cereal and fruit juice to produce ethanol. Do you recollect the metabolic reaction which result in the production of ethanol by yeast? Depending on the type of the raw material used for fermentation and the type of processing with or without distillation, different types of alcohol drinks are obtained. Wine and beer are produced without distillation, whereas whiskey, brandy and rum are produced by distillation of the fermented broth. The photograph of fermented plant is shown in figure. Antibiotics produced by microbes are regarded as one of the most significant discoveries of the 20th century and have greatly contributed towards the welfare of human society. Anti is a Greek word that means against, and bio means life. Together means against life in the context of disease-causing organism, whereas with reference to human being, they are pro-life and not against. Antibiotics are chemical substances which are produced by some microbes and can kill or retard the growth of other disease-causing microbes. You are familiar with the commonly used antibiotic penicillin. Do you know the penicillin was the first antibiotic to be discovered, and it was a chance discovery. Alexander Fleming, while working on Staphylococcus bacteria, once observed a mold growing in one of his unwashed culture plate around which Staphylococcus could not grow. He found out that it was due to the chemical produced by the mold and named it penicillin after the mold Penicillium notatum. However, its full potential as an effective antibiotic was established much later by Ernst Chan and Howard Florey. This antibiotic was extensively used to treat American soldiers wounded in World War II. Fleming, Chen and Florey were awarded the Nobel Prize in 1945 for this discovery. After penicillin, other antibiotics were also purified from other microbes. Can you name some other antibiotics and find out their source? Antibiotics have greatly improved our capacity to treat readily diseases such as plague, whooping cough, diphtheria, and leprosy, which used to kill millions all over the globe. Today, we cannot imagine a world without antibiotics. chemical, enzyme and other bioactive molecules. Microbes are used for commercial and industrial production of certain chemicals like organic acid, alcohol and enzyme. Example of acid producer are Aspergillus niger, a fungus of citric acid, Acetobacter aceti, a bacterium of acetic acid, Clostridium botulism, a bacterium of butyric acid, and Lactobacillus, a bacterium of lactic acid. Yeast Saccharomyces cerevisiae is used for commercial production of ethanol. Microbes are also used for production of enzyme. Lipases are used in detergent formulation and are helpful in removing oily stain from the laundry. You must have noticed that bottled fruit juice brought from the market are clear as compared to those made at home. This is because the bottled juice are clarified by the use of pectinase and protease. Streptokinase produced by the bacterium Streptococcus and modified by genetic engineering is used as a clot buster for removing clots from the blood vessel of patients who have undergone myocardial infarction leading to heart attack. Another bioactive molecule, cyclosporin A, that is used as an immunosuppressive agent in organ transplant patients, is produced by the fungi Trichoderma polysporium. Statin produced by yeast Monococcus purpureus have been commercialized as blood cholesterol lowering agent. It acts by competitively inhibiting the enzyme responsible for synthesis of cholesterol. Microbes in sewage treatment. We know that large quantities of waste water are generated every day in cities and towns. A major component of this waste water is human excreta. This municipal waste water is also called sewage. It contains large amount of organic matter and microbes, many of which are pathogenic. Have you ever wondered where this huge quantity of sewage or urban waste water is disposed of daily? This cannot be discharged into natural water bodies like rivers and streams directly. You can understand why. Before disposal, hence, sewage is treated as sewage treatment plant (STPs) to make it less polluting. Treatment of wastewater is done by heterotrophic microbes naturally present in the sewage. This treatment is carried out in two stages. Primary treatment. This treatment step basically involves physical removal of particles large and small from the sewage through filtration and sedimentation. These are removed in stage. Initially, floating debris is removed by sequential filtration. Then the grit, soil and small pebbles are removed by sedimentation. All solids that settle form the primary sludge, and the supernatant form the effluent. The effluent form from the primary settling tank is taken for secondary treatment or biological treatment. The primary effluent is passed into large aeration tank where it is constantly agitated mechanically and air is pumped into it. This allows vigorous growth of useful aerobic microbes into flocks, masses of bacteria associated with fungal filament to form niche-like structure. While growing, these microbes consume the major part of the organic matter in the effluent. This significantly reduces the BOD, biochemical oxygen demand of the effluent. BOD referred to the amount of the oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by bacteria. The sewage water is treated till the BOD is reduced. The BOD test measures the rate of uptake of oxygen by microorganisms in a sample of water, and thus indirectly BOD is a measure of the organic matter present in the water. The greater the BOD of wastewater, more is its polluting potential. Once the BOD of sewage or wastewater is reduced significantly, the effluent is then passed into settling tank where the bacterial flocks are allowed to sediment. This sediment is called activated sludge. A small part of the activated sludge is pumped back into the aeration tank to serve as inoculum. The remaining major part of the sludge is pumped into large tank called anaerobic sludge digester. Here, other kind of bacteria which grow anaerobically digest the bacteria and the fungi in the sludge. During digestion, bacteria produce a mixture of gases such as methane, hydrogen sulfide and carbon dioxide. These gases form biogas and can be used as source of energy as it is inflammable. The effluent from the secondary treatment plant is generally released into natural water bodies like rivers and streams. An aerial view of such plant is shown in figure. You can appreciate how microbes play a major role in treating millions of gallons of wastewater every day across the globe. This methodology has been practiced for more than a century now in almost all parts of the world. Till date, no man-made technology has been able to rival the microbial treatment of sewage. You are aware that due to increasing urbanization, sewage is being produced in much larger quantity than ever before. However, the number of sewage treatment plants has not increased enough to treat such large quantities, so the untreated sewage is often discharged directly into rivers, leading to their pollution and increase in waterborne disease. The Ministry of Environment and Forest has initiated Ganga Action Plan and Jamuna Action Plan to save these major rivers of our country from pollution. Under this plan, it is proposed to build a large number of sewage treatment plants so that only treated sewage may be discharged in the river. A visit to a sewage treatment plant situated in any place near you would be a very interesting and educating experience. Microbes in production of biogas. Biogas is a mixture of gases containing predominantly methane produced by the microbial activity and which may be used as fuel. You have learned that microbes produce different types of gaseous and end product during growth and metabolism. The type of the gas produced depends upon the microbes and the organic substrate they utilize. In the example cited, relation of the fermentation of dough, cheese making and production of beverage, the main gas produced was carbon dioxide. However, certain bacteria which grow anaerobically on cellulosic material produce large amount of methane along with carbon dioxide and hydrogen. These bacteria are collectively called methanogens, and one such common bacterium is Methanobacterium. These bacteria are commonly found in the anaerobic sludge during sewage treatment. These bacteria are also present in rumen, a part of stomach of cattle. A lot of cellulosic material present in the food of cattle is also present in the rumen. In rumen, These bacteria help in the breakdown of cellulose and play an important role in the nutrition of cattle. Do you think we human beings are able to digest the cellulose present in our foods? Thus, the excreta dung of cattle, commonly called gobar, is rich in these bacteria. Dung can be used for generation of biogas, commonly called gobar gas. The biogas plant consists of a concrete tank 10 to 15 feet deep in which biowaste were collected and slurry of dung is fed. A floating cover is placed over the slurry which keeps on rising as the gas is produced in the tank due to the microbial activity. The biogas plant has an outlet which is connected to a pipe to supply biogas to nearby houses. The spent slurry is removed through another outlet and may be used for fertilizer. Cattle dung is available in large quantity in the rural areas where cattle are used for a variety of purposes. So biogas plants are more often built in rural area. The biogas thus produced is used for cooking and lighting. The picture of the biogas plant is shown in figure. The technology of biogas production was developed in India mainly due to the effort of Indian Agriculture Research Institute (IARI) and Khadi and Village Industry Commission (KVIC). If your school is situated in a village or near village, it would be very interesting to inquire if there are any biogas plant nearby. Visit the biogas plant and learn more about it from the people who are actually managing it. Microbes as biocontrol agent. Biocontrol refers to the use of biological method for controlling plant disease and pests. In modern society, this problem has been tackled increasingly by use of chemical, by use of insecticides and pesticides. These chemicals are toxic and extremely harmful to human beings and animals alike and have been polluting our environment, soil, ground, water, fruit, vegetable and crop plant. Our soil is also polluted through our use of weedicide to remove weed. Biological control of pest and disease. In agriculture, there is a method of controlling pests and that relies on natural predation rather than introduce chemical. A key belief of organic farmer is that biodiversity furthers health. The more variety a landscape has, the more sustainable it is. The organic farmer therefore works to create a system where the insects that are sometimes called pests are not eradicated but instead are kept at manageable level by complex system of check and balance within a living vibrant ecosystem. Contrary to the conventional farming practices, which often use chemical method to kill both useful and harmful life forms indiscriminately, this is a holistic approach that seeks to develop an understanding of the web of interaction between the myriad of organisms that constitute the field fauna and flora. The organic farmer holds the view that the eradication of creatures that are often described as pests is not only possible but also undesirable, for without them the beneficial predatory and parasitic insects which depend upon them as food or host would not be able to survive. The use of biocontrol measure will greatly reduce our dependence on toxic chemical and pesticide. And important part of the biological farming approach is to become familiar with various life forms that inhabit the field predator as well as pest, and also their life cycle, patterns of feeding and the inhabitat that they prefer. This will help develop appropriate means of biocontrol. The very familiar beetle with red and black marking, the ladybird and dragonfly, are useful to get rid of aphid and mosquito respectively. An example of microbial biocontrol agent that can be introduced in order to control butterfly caterpillar is the bacteria Bacillus thuringiensis, often written as BT. These are available in sachet as dried spore, which are mixed with water and sprayed onto vulnerable plants such as brassicas and fruit trees, where these are eaten by the insect larvae. In the gut of the larva, the toxin is released and larvae get killed. The bacterial disease will kill the caterpillar but leave other insects unharmed. Because of the development of method of genetic engineering in the last decade or so, the scientists have introduced B. thuringiensis toxin gene into plants. Such plants are resistant to attack by insect pests. BT cotton is one such example, which is being cultivated in some state of our country. You will learn more about chapter 12. A biological control being developed for use in the treatment of plant disease is the fungus Trichoderma. Trichoderma species are free-living fungi that are very common in root ecosystem. They are effective biocontrol agents of several plant pathogens. Baculovirus are pathogens that attract insects and other arthropods. The majority of baculoviruses used as biological control agents are in the genus Nucleopolyhedrovirus. The viruses are excellent candidates for species-specific narrow-spectrum insecticidal application. They have been shown to have no negative impact on plants, mammals, birds, fish, or even non-target insects. This is especially desirable when beneficial insects are being conserved to aid in an overall integrated pest management program, or when an ecological sensitive area is being treated. Microbes as biofertilizer. With our present-day lifestyle, environmental pollution is a major cause of concern. The use of chemical fertilizer to meet the ever-increasing demand of agricultural produce has contributed significantly to this pollution. Of course, we have now realized that there are problems associated with the overuse of chemical fertilizer, and there is a large pressure to switch to organic farming. The use of biofertilizer. Biofertilizer are organisms that enrich the nutrient quality of the soil. The main source of biofertilizer are bacteria, fungi, and cyanobacteria. You have studied about the nodule on the roots of leguminous plant formed by the symbiotic association of rhizobium. These bacteria fix atmospheric nitrogen into organic form, which is used by the plant as nutrient. Other bacteria can fix atmospheric nitrogen while free living in the soil. Example, Azospirillum and Azotobacter, thus enriching the nitrogen content of the soil. Fungi are also known to form symbiotic association with the plant mycorrhiza. Many members of the genus Glomus form mycorrhiza. The fungal symbiont in this association absorbs phosphorus from soil and passes it to the plant. Plant having such associations show other benefits also, such as resistance to root-borne pathogens, tolerance to salinity and drought, and an overall increase in plant growth and development. Can you tell what advantages the fungus derive from this association? Cyanobacteria are autotrophic microbes widely distributed in aquatic and terrestrial environment, many of which can fix atmospheric nitrogen. Example: Anaerobia, nostoc, oscillatoria, etc. In paddy field, cyanobacteria serve as an important biofertilizer. Blue-green algae also add organic matter to the soil and improve its fertility. Currently, in our country, a number of biofertilizers are available commercially in market, and farmers use these regularly in their field to replenish soil nutrients and to reduce dependence on chemical fertilizer.